I want to, there we go, start to talk about who's going to be on this next segment. We'll have our first guest is Lucienne Ronco from the Fulcrum Therapeutics, she's scientist in the biomarker research side of things. We've been talking to Fulcrum earlier when we were talking to them about, we had Robert Gould on, the CEO, and we wanted to bring in a scientist to talk about that side of it. And we bring in Lucienne Ronco in from Fulcrum. Lucienne, I hope I'm pronouncing your name right. This was an ongoing quick discussion before we went live, so I apologize if I've just butchered it.
No, Tim, you didn't butcher it at all. You got it just right. You must have French in your background somewhere.
Oh, maybe. Yeah, I should get that DNA test. That might be more fun to read than the one that said FSHD1. So yeah, well, that's great. Yeah, I think I should. Well, welcome. Thanks for being here and spending some of the Giving Tuesday time with us and all the hard work that you do with Fulcrum. I know a lot of the listeners are obviously very aware of what Fulcrum is as a company and the great work that is being done in working on, getting us to treatment, right? That's always the goal. So why don't we start maybe though at the base here.
Okay.
What's your role at Fulcrum, and what's a day in the life for you?
Great. Thank you. That's a great question. I always think it's good that folks know what scientists are doing day to day and how we really try to engage and understand the disease and the data. I just want to thank you for letting me be a part of your telethon and letting me be able to tell you a bit about what I'm doing and what Fulcrum is doing. I know Robert, our CEO, was on earlier, and we really value the work that the society does and our opportunities to engage with you and to reach the community so we can build awareness of what we're doing and also how we're advancing research to really understand the disease. And so I'm Lucienne Ronco. I am the Vice President, Head of the Translational Medicine group at Fulcrum. And what is translational medicine? In that role, I work to ensure that the work that we do in our preclinical models gets translated well to our clinical plans and our clinical capabilities. And also so that everything we learn from our clinical studies comes back to our preclinical team so that they can understand the clinical disease and thereby providing a glue and an avenue for communication between preclinical and clinical teams so that we make the most of everything that we learn. In the role, I research what we hope will help us to develop a new treatment for FSHD. As you know, we're focusing on the root cause of FSHD. We hope that our therapy reduces DUX4 expression, and my role is to build tools that allows us to measure, does DUX4 go down in skeletal muscle in FSHD patients and FSHD subjects? Also, does MRI or other imaging modalities help us understand the disease? And does it track the disease progression? So basically, we call these things biomarkers of disease, and they're biological markers that let us follow the disease and understand the disease and understand if our new therapy may be effective in FSHD.
What was the driving force to kind of get involved, directly with FSHD?
The driving force was we saw a very strong need because there are no medicines that treat FSHD. I think that was the first. A second driving force is we saw an organized patient group who were willing to engage and willing to work with us. I think third, we saw organized clinicians like Robbie Tewill, like Jeff Statland, who were doing a lot of natural history studies that we could use to inform our clinical trials, and that was very important. Also, when you have a very clear clinical cause of the disease, which is overexpression of DUX4 in this case, we believed that we could generate a hypothesis that if we found a small molecule that could reduce DUX4, we could impact the disease. And that gives us a very clear question to ask in a clinical study. And all of those things together, the preparedness of the community, the preparedness of clinicians like Robbie Twill, and the ability to ask a very focused clinical question took us towards FSHD.
What's the biggest challenge that you're facing within that? I mean, I know definitely, right, the challenge of COVID-19, I'm sure is right there too, but you know, in conjunction with that, right, in addition to.
Yeah. Honestly, COVID was a challenge, but I think we worked amazingly well with PIs of our phase 2 clinical studies and the patients who responded exceptionally well. And if they had to delay a visit to the doctor associated with our clinical study, they delayed, but they came. We had very few subjects, almost all subjects actually were able to participate to the end. So that, while it was a challenge, we were able to overcome that. And that was a real commitment on the part of the subjects and the patients and our physicians who were running the study. The challenge, as I know you know, in FSHD is how heterogeneous this disease is. In each individual, it affects different muscles at different times in different ways. And the effect is seen in most subjects, not all subjects, over a longer period of time. And so one of the challenges to us is how do we measure a response sooner rather than later in a shorter period of time rather than on a long period of time? Do we have biochemical and molecular endpoints we can study? Are there patient endpoints that we can study? And we've invested a lot of time in asking those questions together with Rabi and Jeff and their large consortium of clinicians who are following the natural history of the disease.
Now, when we had Robert on earlier, he talked about the Redux 4 phase 3. Can you expand on what that's about and what's happening within there?
I can tell you that. So it's a phase 2 study, and it's okay. And it's a phase 2 study that's asking about safety, tolerability, and efficacy in FSHD subjects. We have about 80 subjects around the world, in Europe and Canada and in the United States, working in that, participating, sorry, we're working in the study, they're participating, and it's a randomized placebo-controlled study. So if there are 80 people, 40 will get placebo and 40 will get losmapimod. That allows us to compare in a population that meets our criteria to be included in the study. And it allows us to compare those that receive placebo to those that receive losmapimod and ask if at the end of the study there'll be a difference between the two groups. And it's really the state-of-the-art type of approach to understanding if a new small molecule has activity in a disease area. And we're running MRI to follow disease progression in muscles. We're running molecular endpoints that allow us to measure DUX4 in biopsies, and we're running functional assessments. And I know Michelle, you're speaking to her, I think in about an hour, Michelle Melian, our lead clinician who's running the program, could talk to you a bit more about this, but we're running functional assessments, something like, we call it reachable workspace. It measures the the motion and range of motion and extent of motion of the upper arms. The timed up and go that we've modified for FSHD is called the FSHD tug. It measures an individual's ability to sit up from a supine or a laying down position and then get up and then walk and then turn around, come back and lie down again. Really tailored towards FSHD subjects where there are issues with trunk muscle function, and we hope that we can measure that over the course of the study. So I've just given you 2 examples, but we have other outcome assessments that measure the function of muscles, many actually, because, you know, this is the first randomized control clinical trial of this size in FSHD subjects. There's still a lot to learn. And so we run a lot of endpoints, hoping that we will be able to find some informative endpoints.
And is it looking good then as you approach into a phase 3? Is it promising as you move it forward?
So we don't have the data yet. We had an interim analysis that was promising, and we published that data earlier this year. And based on that data, we're continuing the study, and the study should be completed. I believe we actually have our last biopsy and our last MRI is either late at the end of this year or early at the beginning of next year. This data will come out at the end of the second quarter or close to the end of the first half of next year. Okay. And we are planning our phase 3, and that work is work that we're doing in the background as we analyze this data from this phase 2, Redux 4.
Now, is this an obvious question of what's the most exciting part of your job? Is it what you're talking about? Because, the promise of there's promise, that there's positive results even in the beginning stages?
That is a very exciting part. Honestly, I love just learning about the disease. I love meeting patients and understanding from them what's important to them and thinking about how we can take their input to us and turn it into important and useful endpoints in the clinical trial. I also love that, when you find small molecules that seem to have the activity in your preclinical work that could be valuable to patients and to their families, it makes your hair stand up on the back of your neck. I remember the day we found this first result, and I was standing there with another scientist who was key in identifying losmapimod, and we just looked at each other and said, oh my gosh, what an opportunity. So we are really excited to be able to have brought it to a clinical trial.
You feel like you're making history then, would you say?
Well, I think I hope we're going to make value for patients and for their families. It will be history if it's successful, and if it successfully treats subjects and helps with this disease, it will be historic because there's nothing else at this time.
Yeah, absolutely.
Yeah.
We've had comments already, thank you, Fulcrum, for pioneering clinical trials targeting DUX4. So the community is well aware of all the great work that your company and you personally are doing. What is the importance, in your opinion, of the involvement of patients or this FSHD community?
They've taught us a lot. They've taught us what's important to them, what's important functionally. They've answered lots of questionnaires for ourselves and for Rabi and Jeff that have let us understand something about the pain that they suffer, something about the psychological toll. They've also, we ran a preparatory study earlier 2 years ago that lasted into this past year, where we asked patients to give us 2 biopsies, take MRIs and give us 2 biopsies from an affected muscle. And that was a profound commitment on the part of those individuals who said, yes, take my biopsy so that you can prepare for a phase 2 clinical trial. That was very valuable to us, and it followed up to work that the Seattle Wellstone group had done, and that we were able to validate their findings and extend their findings. Something that's very important to Fulcrum, we're doing the best clinical trial that we can. If we're successful, that'll put us in one place, and if it's not successful, we'll be able to publish this information that patients have provided to us by their biopsies, by their commitment to being in the study, and by the responses that they've given to the patient-reported outcome evaluations. And that published information is going to help pave the way for future trials, for future small molecules to be evaluated. And so we realize that the value is immediate to us and to patients, but also it's longer term because we're expanding that body of knowledge about the disease.
Before I let you go, there is actually a question from a listener or watcher, I guess.
Okay.
Helen asks, are the specific physical assessments you mentioned, i.e. the upper arm reach and trunk assessments, available to share with our doctors and therapists?
They have been published, and they are available to share. That's something that I'd have to speak with some of the clinicians that I work with and the clinicians who have established these endpoints, but they're in the public domain and able to be shared. Yep.
Excellent. Thanks, Helen, for asking the question. And thanks, Lucienne, for joining us. As I got it right the first time. I got all tongue-tied.
Sorry.
I really appreciate your time and all the work you're doing, of course. And my voice isn't my own. I know that. I'm speaking for the many that are extremely grateful for everything you do every day. You wake up, you go to work, your job is hugely valued. And we appreciate your job and your time today that you've given us.
Thank you so much. You know, we really do think of patients every day, and I really do. And I'm just really grateful to have been asked to be involved. So thank you again.
You bet. Have a great day. Enjoy your Giving Tuesday.
All right, Tim, take care. Bye-bye.